Welcome back to the genetic series. Today we'll be looking at chromosomal and non-chromosomal sex determination in animals. Now, the nucleus of all cells harbor hereditary materials called chromosome, and there are two types of chromosome. We have the sex chromosome and we have the autosomes that are also known as ordinary chromosomes. The autosomes control various characters in the body, while the sex chromosome is primarily concerned with the determination of sex in animals. Now, all animals have one pair of sex chromosomes. All the remaining are autosomes. So, for example, in man, where we have 23 chromosomes on the male and 23 from female, one pair of sex is sex chromosome, while the remaining is or are autosomes. There are 46 chromosomes in a complete human, so a pair is sex chromosome, meaning that the remaining 44 are autosomes. The sex chromosomes help in determining the sex of the individual animal. So female animals of mammals and insects are homogametic, meaning that they carry XX set of chromosomes, while the males are heterogametic because they carry X and Y set of chromosomes. So this situation is, however, reversed in birds and some species of fishes. In birds, it is the female that are heterogametic, carrying ZW set of chromosomes, while the males are homogametic, carrying ZZ set of chromosomes. So it means that the females will determine sex in birds, while it is the males that determine sex in mammals. Then there are two broad types of sex determination. We have what is known as the chromosomal sex determination, and we also have the non-chromosomal or environmental sex determination. So let's take a look at the chromosomal sex determination first. Here, sex is determined by differences in the sex chromosome that is carried by an individual. So when an individual is XX, it's a different sex. XY is a different sex. As in mammals, ZZ, different sex, and ZW, also a different sex we produce because animals are carrying these chromosomes. And the sex of the embryo is therefore fixed at the point of fertilization, where either of the parents contribute a type of chromosome towards determining the sex of the individual. The X and Y bearing chromosomes carried on the spermatozoa are produced in fairly equal number or ratio, to say a 50-50 ratio or 1:1 ratio. So they have an equal chance of fertilizing an egg. Therefore, at fertilization, there's an almost equal ratio of male and female embryos that will be formed. and this is termed or known as the primary sex ratio. The implication is that the embryo has an equal opportunity or potential for sexual differentiation. It can either differentiate into a male or differentiate into a female, and this is because they have two sets of primitive genital tracts that are known as the Wolffian duct and the Mullerian duct. So at 6 weeks, sexually indifferent is known as the sexually indifferent stage. So at 6 weeks, these are the primitive gonads, and this can differentiate either into the Mullerian duct or the Wolffian duct. If it differentiates into the Wolffian duct, then it will develop, the embryo will develop eventually having epidermis, the vas difference, the seminal vesicles, as well as the ejaculatory duct, meaning this will develop into a male. Or if differentiation is toward the Mullerian, then the embryo will develop the fallopian tubes, uterus, and also the upper part of the vagina. Normally, and this is under the influence of sex chromosome, only one set of these ducts develop post-fertilization, while the other one will slowly regress or become residual and stop development. So while the Mullerian duct will develop into a female, as said earlier, the Wolffian duct will develop into a male. And this development of the Mullerian duct, particularly into a female, is, however, not because the embryo possesses the X and X chromosome, but rather it is a permissive state in the absence of the Y chromosome, meaning that maleness is the dominant state, and it is only the absence of the Y chromosome, and specifically the absence of the male sex hormone from the embryonic testis, that will cause the Mullerian duct to become more prominent, while the Wolffian duct will regress. So the growth of the Mullerian duct will continue if or when the Wolffian duct regresses, and then the embryo can, at this stage, be clearly distinguished as a female. So this is shown in this graph, in this figure here, in this figure here. So if we have a male having X and Y chromosome, and this is a particular uh, gene that we'll be looking at later. So the embryo can either become a male, the gonads will produce testes, but in the absence of the Wolffian duct developing, then the Mullerian duct will develop. So then the gonads will produce a testis, and this will go on into the Leydig cells, will produce testosterone. The Wolffian duct will continue to develop and give rise to the epidermis, vas difference, seminal vesicle, and the ejaculatory duct. And we see that if the testis does not develop, then the routine will go the other way and we produce the Sertoli cells, uh, and then the Mullerian duct will slowly degenerate. So if the embryo is XY, as seen in the last pictorial, the SRY gene, which is the sex determining region of the Y chromosome, will be present, and it will produce a set of protein which will activate a gene network that will direct the gonads to develop as testes. And then the two support cells in the testes will then differentiate and they'll begin to generate important regulatory molecules. And these molecules will direct sexual differentiation. Therefore, the Leydig cells, which produces testosterone, will promote the development of the Wolffian ducts. So the Wolffian ducts then will differentiate and form the epidermis, the vas deferens, the seminal vesicles, and the ejaculatory ducts. And the Sertolic cell, on the other hand, will produce the malaria inhibiting substance. This is also known as MIS or anti-malarial hormone AMH. This is a peptide hormone which will cause the malaria, that's the female duct, to regress and stop development. But in the absence of a Y chromosome and the SRY gene, however, this situation the gonads will develop as ovaries. So, and then because there will be no Sertolic cells and no influence of the MIS hormone, the Mullerian duct will continue to develop and then it will produce the fallopian tubes, uterus, and other parts of the female reproductive organs. So in this case, there will be no Leydig cells produced and then no testosterone will be produced. As a result of that, the Wolffian duct will slowly degenerate. So female development will proceed in this situation in the absence of the SRY gene. And this is because of no production of testosterone or the malaria inhibiting substance. So the Wolffian duct in this case will continue will regress while the Mullerian duct will continue to developing into the fallopian tube, the uterus, and the upper part of the vagina. So the key factor in determining sex actually is the role that is played by the Y chromosome, which contains the SRY gene. So the SRY is linked to maleness as well as the secretion of the male sex hormone by the embryonic testes. So let's look at other variants of chromosomal sex determination. In the fish, Zygiophorus varietus, in addition to the normal XY presentation of the sex chromosome, there is found also another format in which fish with XY chromosomes are female, while those with YY chromosomes are males. This is a direct opposite of what is expected. And then there's also the XX and XO sex determination, which is found in some rats, some mouse, seeds, and some insects, such as the ant. In this format, the Y chromosome is found to be completely missing. As a result, the females have XX chromosome, while the males have XO chromosomes. So in this case, it is the females that will determine the sex of the animal or the insect. In birds, as we've seen earlier, and in most reptiles and in some fish species, the normal system of sex determination is the ZW chromosome system. So here it is the female that are ZW and heterogametic, while the males are ZZ and homogametic. Therefore, it is the females that will determine the sex of the, the bird. In bees and ants, fertilization determines the sex of the progeny. So if the egg is fertilized, it will develop into a female. And if it is not fertilized, it will develop into a haploid male, which does not have a father. So this haploid male does not reproduce sons, and this process is known as parthenogenesis. So we want to look at non-chromosomal sex determination quickly. In some organisms, sex of the individual is determined by environmental factors. In the turtle, alligator, the Australian lizard, for example, the sex is determined by the incubation temperature. So if the eggs are incubated under high temperature, they develop into males, while if they are incubated under low temperature, they will develop into females. Chemicals also have effect on the determination of sex in some organisms. In the marine worms, for example, it is the larva becomes male if they make contact with a chemical called gonadal, and this chemical is produced by the female marine worm. Those larvae that are found at the bottom of the ocean, however, develop into females. In the clownfish, physical activity affects the sex of the progeny. So here the dominant individuals, the most active in the group, develop into female, while the less active and the less dominant develop into males. Sex reversal is also possible. This has been experienced in animals such as in snails and fish, and we can see that the determination of sex has become very important in livestock production. And this is particularly so where the farmer is interested in a particular product, and the product can only be produced by a particular sex of the animal. For example, when the farmer is interested in meat, in fish, meatiness in fish, for example, or egg production in poultry beds, and also meat production in cattle, then the determination of the sex at an early stage becomes very primary, and the farmer is very interested in knowing the sex of the organism even before they are born. And if there's any way that this can be influenced, it will be of particular importance to the farmer, hence things like sex reversal. For meat and fish production, particularly, males are usually the most preferred, and this is because they have faster growth rate. So thank you for listening, and we've come to this brief lecture on sex determination in animals.